Dear students, today we will be discussing mechanism of nutrient uptake in plants. The objective of our study would be to enlist the various essential mineral nutrients and depict the main chemical forms in which these mineral elements are acquired by plants, and to understand the mechanism of nutrient uptake in plants. As you know, plant performance depends on the availability of sufficient quantities of essential mineral elements. There are 14 essential mineral elements required for plant growth and development. These include the macronutrients like nitrogen, phosphorus, potassium, calcium, magnesium and sulfur, which are required in large amounts by plants. The micronutrients on the other hand are chlorine, boron, iron, manganese, copper, zinc, nickel and molybdenum, which are required in smaller amounts. Deficiency in any one of these elements restricts plant growth and reduces crop yield. Table 1 depicts the main chemical forms in which mineral elements are acquired from the soil solution by roots and the critical leaf concentrations for their sufficiency and toxicity in non-tolerant plants. The critical concentration for sufficiency is defined as the concentration in a diagnostic tissue that allows a crop to achieve 90% of its maximum yield. The critical concentration for toxicity is defined as the concentration in a diagnostic tissue above which yield is decreased by more than 10%. It should be recognized that critical tissue concentrations depend upon the exact solute composition of the soil solution and can differ greatly both between and within plant species. The later differences reflect both ancestral habitats and ecological strategies. Most of these elements are found in soil solution either in the form of inorganic or organic salts or ions, which may exist either in free state or bound to clay particles. Whenever there is depletion of any free ions from the soil solution, respective ions are released from clay particles into the soil solution to maintain the equilibrium. This is achieved by a process called ion exchange process. This may be due to contact ion exchange mechanism or by carbonic acid ion exchange mechanism. Now, let us discuss contact ion exchange mechanism first. Plant roots are in contact with soil clay particulates which have colloidal dimensions. Plant cells which are living secrete hydrogen ions which are positively charged. Such ions can easily displace cations like potassium and sodium ions that are bound to clay particulates. Thus, the cations are made available for the root system to absorb the required ion. Both the relative retentive capacity of the clay particles, though not absolutely fixed, is in the order of hydrogen is greater than calcium, is greater than magnesium, is greater than potassium, is greater than ammonia, and which is in turn greater than sodium. Thus, hydrogen ions are capable of replacing any cation found on the clay particles. Nonetheless, it appears that hydrogen ions can replace any bound ions to clay particles, but there is a preference of ions to be released. Now, let us discuss carbonic acid ion exchange mechanism. As you know, roots continuously respire irrespective of day or night and liberate significant quantities of carbon dioxide, which when dissolved in soil water produce carbonic acids. Immediately they ionize into hydrogen and bicarbonate ion. The hydrogen ions thus formed are capable of exchanging with any bound cation on clay particles and make the bound cations available for the roots. Though the above mechanisms were once proposed as theories, it is now clear that growing roots not only release carbon dioxide but also secrete hydrogen ions. Thus, the root does provide hydrogen ions for both carbonic acid ion and contact ion exchange processes. Similarly, adsorbed anions are also exchanged by anions like hydroxyl ion. Now, let us see structures involved in mineral absorption. Aquatic plants do not need any special structures for the absorption of minerals, for the entire body acts as an absorptive surface. But terrestrial plants possess extensive root system with innumerable growing apices. Short-term radioactive isotope labeling experiments indicate that the meristematic regions of the root absorbs greater amount of ions than any other region. This is perhaps necessitated by their active metabolic state. Though most of the minerals are absorbed by the growing meristems, minerals ultimately find their way into the xylem elements by active transport. From the xylem elements, they move upwards along with the transpiration stream and get distributed to all other regions of the plant. Now, let us study the actual mechanism of absorption. From time to time, various theories have been proposed to explain the mechanism of ion uptake or absorption. Salt respiration theory by Lundergaard, cation ladder theory by Middleton and Russell, lecithin carrier hypothesis by Bennett, Clark, and others have made attempts to explain the mechanism, but all of them fail to explain experimental observation. Though the most theories have no relevance to what we know today, these theories have been retained in many textbooks as historical landmarks in understanding the mechanism. As evidenced by experimental observations, the overall process is not a single mechanism, but is a combination action of multiple events. Without going into the details of various events, a comprehensive account will be given in this presentation. First, let us discuss the passive phase. When the intact root system is provided with exogenous supply of mineral nutrients, to begin with, ions diffuse into the apparent free space found within the cell wall and the intercellular spaces found in the tissue. The rate of diffusion depends upon the steepness of ionic gradient between the external solution and the solution found in apparent free space. In fact, the fine microspaces found in the cell walls never act as impediment for the free diffusion of ions. Instead, the charged cellulose material greatly facilitates rapid diffusion of ion into the cell wall spaces. Thus, ions diffuse through the apparent free space of the root system and cross through the cortical cells and outer xylem elements through the passage cells. In xylem cells, the concentration of ions is always low because of continuous upward movement of sap by transpiration pull. So, the initial uptake is very rapid and a passive process. Moreover, this entire event is a kind of mass movement of ions along the concentration gradient till they reach the plasma membrane. It is not just mineral uptake is facilitated by protein transporter, even water is carried by their own protein carriers called aquaporins. They have transcellular domains at one surface they have water binding site. When water binds, it changes its structure in such a way water is released at the outer side. Now, let us discuss the active phase. Once the apparent free space are filled with mineral nutrients by free diffusion, the entry of ions across the plasma membrane into the cellular cytoplasm is highly regulated. In this sense, the uptake is selective and energy dependent. Though some ions, because of local destabilization of membrane potential or due to membrane leakiness, diffuse across the membrane along the concentration gradient, but the entry of majority of ions is selective, facilitated and dependent on metabolic energy. Probably that might be the reason as to why most of the ions absorbed by root system are found in meristematic cells, because they are actively metabolic and produce more of ATPs than any other cells. Electron microscope studies of fractured and intact plasma membrane surfaces of plant, animal and bacterial origin reveal that the membranes contain an array of globular proteins of different sizes and dimensions, scanning the entire cross section or a part of the membrane. Some of them are aggregated in one site and others are randomly distributed. However, the position of such granules in the membranes is never constant because of the fluid nature of the membrane. A large number of proteins with their 3D globular structures act as carrier proteins or transport proteins. Such carrier proteins have high affinity towards specific ions because they do contain specific binding site or sites. In the past 10 to 15 years or so, quite a number of carrier proteins have been isolated and purified to homogeneity. The 3D structure of a few of these proteins have been determined. Some of the common examples for carrier proteins are potassium, sodium ATPase pumps, hydrogen protein pumps, calcium transporting protein called calmodulin, phosphate carrier proteins, etc. The above mentioned proteins have been isolated from different sources and their structures as well as kinetin properties have been studied in detail. Interestingly enough, the discovery of some of cyclic polypeptides like valinomycin, nystatin, cyclosporine, etc. from many lower fungi have given a new dimension to the carrier mediated ion or nutrient uptake. Such compounds are called ionophores because when they are added to the medium, they penetrate into the membrane easily and create a hole which permits the entry of only one specific kind of ions. For example, valinomycin greatly facilitates the uptake of potassium ions only. Similarly, nystatin allows specific anions. It is now speculated that the plasma membrane contains such specific ion transporting complexes and they are responsible for the movement of certain ions under certain circumstances. Ionophores' role can be used as a generalized process of ion uptake, but they too play a significant role in the entry of specific ions in some species. Using mutation as the tool, molecular biologists have discovered that there are specific genes for specific carrier proteins which transport a specific substance. and such carrier proteins have been detected in plasma membrane of the cell. Inorganic ions, monosaccharides, amino acids and many such components including proteins and nucleic acids have been found to be transported across membrane facilitated by specific membrane proteins. Some of the receptor proteins have been found to carry the stimulants across the membranes. Most of the experimental evidences suggest that the ion uptake is carrier mediated and the carriers are proteins. Only proteins can explain certain properties like ion antagonism, specificity of ion uptake, saturation effect, kinetic properties, requirement of metabolic energy, etc. The carrier proteins exhibit 3D shape. They have specific binding sites for the ions or substrates and no other ion can bind to such sites. After binding, the proteins undergo rapid conformational changes in their 3D structure and exhibit mobility in the lipid core of the membrane. For its active transformation and transport, they require metabolic energy. The activity of carrier proteins can be regulated by the effectors or effectors, similar to that of allosteric enzymes. The transport activity of these proteins can be inhibited by competitive or non-competitive inhibitors. Another important feature of carrier proteins is their ability to transport molecules against concentration gradient by active process. Peter Mitchell, a Nobel laureate, has proposed 3 possible mechanisms by which carrier proteins operate in the transportation of ions or other nutrients, namely uniport, antiport and symport mechanisms. Now, let us discuss uniport mechanisms. In this case, the activated carrier proteins pick up a specific nutrient at one surface and translocate across and unload the same on the other side. After unloading, the carrier protein reverts back to its position by conformational change. Many bacterial and animal cells have ATPase dependent carrier proteins which transport many nutrients like glucose and uniport mechanism. On the contrary, the concentration of sodium ions is always higher. There are many such carrier proteins which transport nutrients passively but along with the concentration gradient. Such a process is called facilitated transport, but it is passive. Such uniport mechanism has been observed with respect to the transport of many monosaccharides, amino acids, etc. A very good example for this is calcium binding protein. Now, let us see the antiport mechanism. This process involves coupled transport where one specific ion is transported in and the other is transported out by the same transporter. The antiport mechanism is very well exhibited by sodium potassium ATPase pump found in the plant cells like Nightella and in animal cells like erythrocytes. The internal concentration of potassium ions is manifold higher than the concentration of potassium ions found in the extracellular fluid. Cells have to maintain such high concentrations of potassium ions intracellularly because they are required for various metabolic activities and for the maintenance of turgidity of the cell. On the contrary, the concentration of sodium ions is always higher in the external fluid than inside the cell. Hence, sodium ion often leaks in or taken in by other processes. As excess sodium ions inside the cell is toxic and the same are expelled out. Sodium potassium ATPase pump does this by Antiport mechanism. Now, let us discuss the symport mechanism. Transport of this kind involves the binding of two different molecules or ions to two active sites of the same carrier protein. By virtue of the binding of both the ions, the carrier complex rotates by conformational change. Thereby, the said ions are released simultaneously at the opposite side of the membrane. Such symport type of transportation has been recorded in a variety of organisms, but the best example for symport mechanism is the transport of sodium and glucose found in animal and bacterial cells. In this case, the carrier protein is a complex macromolecule and possesses two sites, one for sodium ion and the other for glucose. While the binding sites of the protein are facing the external fluid, both sodium and glucose bind to their specific sites. The binding is facilitated in the sense the binding of one molecule promotes the binding of the other. However, as soon as sodium and glucose bind to the protein, it undergoes conformational change and opens the inner surface, and the sodium and glucose are released in the cytoplasm almost at the same time. The release of molecules makes the protein revert to its original shape and position. The sodium ions that enter into the cell are expelled by sodium potassium ATPase pump, that is by Antiport mechanism. With this, we conclude our today's topic. Thank you for your attention.